Very good. Thank you very much. It's interesting. I was reflecting on talking at this, and I thought you really know that the end is nigh when you're asked not to talk about your work but about your career. So with that, I thought, well, how do I lead into this? I was taken actually by a lecture that Sir Peter Medawar gave in 1964. And if you'll bear with me, I'd like to just read a bit from his lecture. And his title was, "Is the Scientific Paper a Fraud?" And this is what he said. He said, "Is the scientific paper a fraud? I do not, of course, mean that the scientific paper misrepresents facts. I mean the scientific paper may be a fraud because it misrepresents the process of thought that accompanied or gave rise to the work described in the paper. Just consider for a moment the traditional form of a scientific paper. First, there's a section called the introduction, in which you describe the general field in which your scientific talents are going to be exercised, followed by a section called previous work, in which you concede more or less graciously that others have dimly groped towards the fundamental truth that you are now about to expand. Then there's a section on methods, and that's okay. Then comes a section called results. This section consists of a stream of factual information in which it's considered extremely bad form to discuss the significance of the results you're getting. You pretend firmly that your mind is a virgin receptacle, an empty vessel for information which floods into it from the external world from which you yourself have revealed. You reserve all appraisal of the scientific evidence until the discussion. And here you adopt the ludicrous pretense of asking yourself if the information you have collected actually means anything. Of course, asking yourself if any general truths are going to emerge from the contemplation of the evidence you brandished in the section called results. And he goes on to say the conception underlying this style of scientific writing is that scientific discovery is an inductive process. Out of a disorderly array of facts, an orderly theory, an orderly general statement will somehow emerge. So he says the scientific paper is a fraud in the sense that it gives a misleading narrative of the process of thought that go into making scientific discoveries. And scientists should not be ashamed to admit that the hypotheses appear in their mind along uncharted byways of thought. So why am I telling you this? I'm telling you this because as I started to look at how my career might look as I lay it out, I thought some people might assume that there was a plan, and there was no plan. And other people might assume there was a random series of events which I've now pulled into some sort of discussion, and it wasn't quite either of those things. In fact, what there was a series of, if you like, hypotheses, things that I thought I was interested in, and I followed. And here perhaps I'll finish this bit with another quote from Peter Medawar, because I've said that actually, certainly for me, my career has been about following things I've been interested in, following the hypothesis of something. So he says, "I cannot give any scientist of any age better advice than this. The intensity of the conviction that a hypothesis is true has no bearing on whether it is true or not." And of course, that's of course totally correct, and applies to some of the things I thought about my career as well. So let me tell you a little bit about my career path, some of the influences, some of the things that I think I've learned, and some of the things that I would think about if I were to start again, although I suspect I couldn't start again because I know from having interviewed a lot for medical schools, I doubt I'd get in these days. So I know that you've got a much better starting substrate than I had. So I trained in medicine at St George's, and my early influences undoubtedly were my teachers. No question about that. And particularly those teachers who integrated science and understanding into medical practice. And so my first point is this. It's been fashionable, it has been fashionable, to suggest that as a clinical academic, you can do 2 out of 3 things: teaching, clinical practice, and research. If you follow that logic, it tells you that you could never be taught on the ward by somebody who's research active, and that can't possibly be correct. And so my first point is that integration of clinical work, teaching and research, I believe is important for academia, and I believe that it's not, of course, the case that everyone does all three equally, but I know from my career that the influence that's come to me from people who've been teachers, researchers and clinicians has been profound, and I do think there's something about teaching which is an important part of the mission, and I will come back to that as I go through. I was lucky enough to do an intercalated BSc and got some exposure to some fantastic lecturers, and I'm sure this will resonate with some of you. Some of those individuals who really lecture about something profound, important and transformational can have a lasting impact. And I thought about doing an intercalated PhD at the time, and there was no such thing at the time, and in fact many people who did that never came back to clinical medicine in those days. So I didn't do it, and I've often wondered how that might have changed my career had I done so, and I am a supporter of the intercalated PhD programme, at least for some people who I think it gives a real opportunity to understand research impact early in a clinical career. I did have another influence, and I'll give you a quote this time from Thomas Lauder Brunton, actually from 1879, and this was quite important for me in my thinking. He said, it wasn't around the time I trained, it was a bit earlier than that, before therapeutics can become a science, the physician must know the action of his drugs, just as a locksmith does of his keys. And since pharmacology is so young, it's a little wonder that medicine is yet only an art. And I have to say, I was completely inspired by pharmacology. I was completely inspired with understanding how you could use chemistry and chemical probes to unlock mechanisms of disease, and that was from quite an early time. I qualified in 1984 from St George's, and then entered the hurly-burly of SHO jobs on what was known as the circuit at the time. And I reflect sometimes on those times of 6-month SHO jobs, which meant that you were applying for a new job about 4 months into, or 3 months into the job you already had. And of course that had some downsides, and things are more structured now. It had a lot of upsides as well, and I think there was something about flexibility there which was quite important, and flexibility is a theme that I'll come back to. And you know, I think actually there was a lot of fun going around that circuit at the time, and one met a lot of very inspirational people. At the end of that, I decided to apply for a lecturer job in clinical pharmacology back at St George's. So my second point is follow your nose. And what I mean by that is I could not have been more strongly advised by every single clinician I asked not to do this job. I said, why on earth would you want to go and do something in clinical pharmacology, and why would you want to go back to St George's? And the reason was I was interested in doing research. The job offered a good clinical content, and perhaps more importantly, I was very inspired by the person who led that unit at the time. And that leads to my point 3, and it's really been mentioned by John in terms of what he's described from the academy. You need a mentor. There's no question that we all need people who can help us, guide us and advise us. And my decision was made largely because the supervisor, who was Joe Collier, was not only an inspiring teacher, but actually spent an inordinate amount of time with me on research. Actually, an embarrassing amount of time when I think back at it, and how much time sometimes I've been able to spend with people. He helped me not only to understand what I wanted to do in research, but actually I think spent a long time talking about research process, research philosophy, and what it was that it meant to be a researcher. He challenged me to think big, and I think that's important. I think it's often easy to become technical and to become limited in terms of the scope of the question you're asking, and I think it's important to be shown a landscape in which you can think big. He was challenging, he was a broad thinker. He took care, actually, and this is important, both to empower me and to push me into areas where I might feel uncomfortable, and to make sure I met people, gave talks. He taught me to think, frankly. He taught me to write, and he taught me to lecture. And I think those things are important. I think they're part of an accountability that we all have as we go through our careers, to make sure that we do take time to do that for the people coming on behind us. After my thesis, I gained MRCP and got my thesis, and then went on to what at the time was called a senior registrar, and I got pulled into sort of slightly more mainstream clinical work and to some extent slightly more mainstream research, I would say. And I realised that I wanted to really have quite a significant research component to my clinical career, but I definitely wanted to continue being a clinician. And I applied for something at the time, which I think I'm right, others may remember, but I think it was called actually the Clinician Scientist Training Scheme. And it was, there was one run by the MRC and one run by the Wellcome Trust, and they were, I think, 7-year training schemes that took you through with a mix of clinical and research-based study right up to senior lecturer level. Having got one of these, I then thought, well, where am I really going to do it? And I made a decision which was absolutely important for everything that happened subsequently in terms of how I thought about research. I made a decision not to spend the entire time of my research in the university sector, but to go and spend time doing a postdoctoral fellowship as part of this scheme in the laboratory of Dr. Salvador Moncada, who was then head of research at the Wellcome Foundation. Now, just because many of you may not remember or know the Wellcome Foundation was the drug company from which the Wellcome Trust was funded. So the Wellcome Foundation was the initial company which Henry Wellcome founded, and then he left in his will the money for the Wellcome Trust. And in fact, when the Wellcome Foundation eventually got bought, the money from that went to create the huge trust fund that now is the Wellcome Trust. So it was a pharmaceutical company, and its research base was in Beckenham. And I did a postdoctoral year in that laboratory, and it was an incredibly exciting lab at the time. It was a lab that was buzzing. It was an academic base, really, in the middle of a research-based pharmaceutical company. It was his personal laboratory of 10 people in the middle of the company. It was a rather odd thing. I think there were about 10 of us. They were from all around the world. So we had people from the US, Canada, Australia, Spain, and I travelled from Tooting, so I was the local one in this. And we were all undertaking work on endothelial biology and nitric oxide signalling, as it happens at the time. Very, very exciting, but probably the biggest eye-opener for me was to understand how to apply a multidisciplinary approach to answering questions. I suddenly moved from an institution where the questions I asked were bounded largely by the techniques I knew in my laboratory, to one where the questions I asked could be answered by a range of technologies which were available across the company. So I started to apply chemistry to biology. I was able to access easily advanced analytical techniques. At the time, things like mass spec and NMR were not widely available across academia, yet there they were, properly staffed, properly maintained, easy to access. Access in vivo models that were really run to a very high quality, and to access a number of different approaches to answer the question. And for me, this was a transformational moment when I realized that the job of a clinical academic was to ask the right question and then find the technologies to answer that question, not to be bound only by the technologies that you happen to understand. Now, of course, it's important that there are individuals who are really expert in technologies, otherwise you can't do that. But I think there are questions which go beyond the limitations of what you may or may not be able to do in a single laboratory. In 1990, I applied for a senior lecturer, a consultant post at St George's, and at the time I was told, and this may resonate with some of you, I was told, well, I don't think you're clinically experienced enough yet, and we want to have you do a period on the wards and then a period off the wards. That was exactly what I wanted. It was music to my ears, and in fact, I think I did 6 months on and 6 months off, which was not that common in those days. I think it was much more common to try and do everything concurrently. And I have to say, I fiercely protected the fact that I had time on and off the wards. And for me, it meant that I could do time on the wards and really concentrate on what the primary objective was there, which was to look after patients and to be a good clinician, and to have time off the wards when I could really spend time concentrating on my research. It does have consequences, and I remember very clearly that a consultant at a neighbouring hospital, when discussing me with one of the registrars at the time, referred to me as a rat doctor. So you have to be resilient in academic medicine because these things will come your way. But the fact was I was doing actually a lot of clinical work at the time and doing acute intake, which for me again was important and actually hugely enjoyable. In 1995 I moved to UCL to take up the chair of clinical pharmacology, and that was part of a project which didn't really succeed. And the project was at the time called the Cruciform Project, and in fact I went there and 4 or 5 others did, some from industry, some from other parts of academia. And the idea was to try to build a new model of interaction within academia. It was a totally academic enterprise, but it brought people in from industry, it brought chemistry in, it brought the prospect of trying to do something which had some practical output in terms of molecules and in terms of progression into the clinic. So I think the idea was broadly right, and actually you can see it in many places now as a concept, this much closer interaction and this building on the strengths of the two sides to really achieve what you want to achieve. So why did it fail? It didn't fail completely. I mean, it ultimately did end up as an institute. I wasn't part of it. I left. I had a difficult time during that. I have to say it was probably the most difficult time of my career. Why did it not succeed? I think it failed because of leadership. And leadership is a point I want to come back to, which I think is underdeveloped as a training skill in academia, and something which I've certainly learned a lot about in my time in industry. It didn't work, and once again I was extraordinarily grateful for advice, support, mentoring from individuals who could take a dispassionate view and actually help me see that this wasn't the first time this sort of hiccup had occurred in a career, and actually there were ways through this and how to manage it. So I'm sure that happens to many people. I can't overemphasize the importance of having those sorts of contacts, those sorts of people you can speak to, and I also can't overemphasize the responsibility that I think we all have to do the same for others. Something else happened around that time. I actually got lots of grants, and my research group expanded very rapidly. And I remember very clearly at the end of a year where I thought we'd been successful. And I thought we'd been successful because I'd brought in money and my research group had expanded. And I sat down at the end of the year and tried to look at what we'd really done. And I'd moved up to, I don't know, let's say 12 people in the lab. Previously, I might have had 3 or 4. And with a horrified look, I realized that there was no way we were 3 or 4 times as productive as when I had 3 people in the lab. We maybe had been 1.2 times as productive. And I got thinking, why was that? And the answer was failure of leadership. I had completely failed to understand the complexities of running a laboratory of 12 people versus the day-to-day interaction with 1 or 2. And all of you will face that, and you'll have your own ways of thinking about it. But I have to say that for me, the time taken to understand what it is people are doing, why they're doing it, how it fits into what you think the lab is about is important. Not to control, because actually I think you want the brightest minds following their own ideas. But what I found was that during that year, people had been busy. Everyone had been busy. Because I'd employed really super smart people who wanted to do great things and wanted to be busy. So there's no question everyone was busy. There's no question everyone was burning through consumables. There's no question that we could spend all the grant money, but we simply hadn't taken the time, I hadn't taken the time to sit down and really make sure that individuals knew what it was that we were trying to achieve and how they contributed to that and what it was that they needed to do to be successful in terms of research. It was a massive, massive eye-opener for me about the importance of leadership communication in laboratories, particularly as your group expands. And in fact, after that, when friends and colleagues would say things like, I've got 25 people in my lab, my first reaction was often, well, how the hell would you know that? Because you wouldn't know from the output sometimes. And the number of people is not the issue, the question is what you're doing. In 2002, I became head of Division of Medicine at UCL, and of course that comes with gradually more administrative work as well as research. And I continued with all of the things, but my lesson there that I took very clearly is you can't do everything. I mean, you can't, and something has to give, and you have to make choices. And for me, one of the things I learned there was that there is a danger if you become extremely overloaded with things, and this was the danger for me, that sometimes really, really important things, like the patient you're looking after, may not get the care and attention that that patient absolutely demands and deserves from you. And so I think you have to make choices and you have to decide what you are going to do and what you're not going to do. And around that time, I also had the pleasure, which I think is one of the real lasting pleasures of a career in clinical academia, of seeing some of the individuals who trained with me become very successful, more successful than me in their own right. And I think that is one of the great pleasures of having a career in academic medicine. So let me try and make some sense of this. Did I have a plan? Absolutely not. Did I know what I was interested in? Yes, I think I did know what I was interested in, and I think I did know the things that excited me and interested me. I also think that some people find the timescale of research extremely disconcerting as a clinician, and I've seen this in my lab. People, on a clinical day, you get instant gratification and instant depression as your day goes on, depending on what's happening. In research, your gratification and despair may be over many, many months. You may see nothing for a long time, and some people just find that impossible, and I think they need a lot of help. And so that's one of the things that I think you need to be careful of as you think about which clinicians really, really want to do research and stay with it. So I knew I wanted to do that. I had a passion for research and a passion for being a clinician. I absolutely benefited from having mentors, one or two or more, and people that I could turn to for advice. There's no question I needed that. I absolutely benefited from having role models. There's no question that there's luck involved in all of this. You get luck and you take it, and there's no question for me that some of the decisions that I made were really driven away from the mainstream as a consequence of knowing what I wanted to do. And so my first message really is around the nature of serendipity in all of this and taking chances. And actually, this is more difficult these days. I believe in a more structured, a more rigid training scheme. I know it's more difficult to do some of the things that I've talked about, but I do believe it's important to take risks. I do believe it's important not to always follow the obvious advice and to sometimes take chances to do something that you really want to do because you believe in it. So let me move now to the next thing, which is curiosity. And this, of course, is absolutely essential for a career in academic medicine, or perhaps more correctly, a career as a clinician scientist, whether that's in academia or in industry. And I don't need to tell you that, because if you look around today and the topics of the posters and the presentations, curiosity clearly underpins a lot of what's going on and what you're interested in. I do think that one of the big advantages of being a clinician and a scientist is, and one of the responsibilities is to be curious about the patients you see. And I think I'll mention three people here, and there are many, and it's an area that I think I probably wasn't good enough at and would like to probably do differently in terms of being really seriously curious about the patient you have in front of you. And the three people I mentioned, I think Keith Peters gave a lecture entitled Exceptional Matters here at the college, and in that he pointed out and really went through a number of things where astute clinical observation linked to scientific understanding led to really opening up the vistas of possibility, both which led to basic science and led to clinical science. A second example, I haven't followed it very closely recently, but I was quite intrigued a few years ago when Stuart Ralston in Edinburgh told me that a man had come into his clinic, I think age 29 or something, with very, very severe osteoporosis of the type that he really shouldn't have got, and he identified an autoantibody which seemed to be driving that. And that sort of observation of the extreme, I think, is exemplified probably most brilliantly by Steve O'Reilly and the work that he's done in terms of looking at rare childhood diseases and diseases of metabolism and identifying some very fundamental mechanisms of disease and indeed of biology as a result of that. So I think the curiosity of what's happening with patients is crucially important. So let me give you 3 examples where for me, curiosity led to something quite interesting. The first was out of teaching. Early in my lecturing career, drew the short straw as far as I was concerned. We obviously had to cover a range of topics in pharmacology, and the short straw for me was the antifungal, antiviral, and anti-something else lecture, which I knew nothing about. And one of the things about teaching is you better make sure you understand it if you're going to go and teach somebody about it, because they'll sure as hell find out you don't know what you're talking about otherwise. So I went away and read up about that, and as a result of that particular lecture, I became very interested in what the effects of clotrimazole may be in terms of, in fact, nitric oxide signaling from something I read in one of the textbooks, and we did some experiments with that. And that was important for 2 reasons. One is it led to a student who was in my lab at the time, Richard Bogle, winning the, I think it was British Hypertension Society prize that year for his presentation, and because it made us as a laboratory get into molecular studies in quite a big way. The second, in terms of curiosity, is to make sure that if your aim is to treat disease, you actually take it to the logical conclusion and see whether you can treat a disease. And here, it was the desire to get into patients, to understand whether the pathways we were looking at did something in patients. And I remember one day, when we'd worked on something to do with septic shock for a long time, somebody said to me, why on earth are you continuing to explore the minutiae of this rather than asking the question, does this work in a patient? And I think that was, again, an important moment for me to realize that you have to get into the clinic and you have to understand, if your aim is to make a therapy, does it actually do something? And the third curiosity comes from interdisciplinary research, which I've mentioned already. I don't think I've been more interested than when I'm talking to people from other fields, and you suddenly realize how their thought processes work, how they think about approaching a problem, and how actually you can apply your general scientific knowledge to their area and have the most extraordinary dialogue and most extraordinary things come out of it. So those interdisciplinary conversations, I believe, are critical. So in 2006, I left UCL to join GSK as head of drug discovery initially, I'll come on to the question of why. I'd written a lot of negative things about the pharmaceutical industry, and I'll come back to that in just a minute. So why, briefly, did I decide to move? I absolutely believe that medicines have changed medicine more than almost anything. I look at examples like the curing of certain types of childhood leukemia. I look at the example of AIDS moving from an automatic death sentence to being a chronic disease by the action of the discovery of antiretroviral agents. I look at the examples from years ago of ulcer wards completely closing because of the ability to treat peptic ulcer disease with drugs, and there are many other examples, and I do believe we're on the era of a new wave of medicines that are going to dramatically change the way we practice medicine. Even medicines that perhaps don't have a massive effect change medical practice enormously, and I think the example that we saw 10 years ago, or maybe a little longer, is the impact of having some sort of therapeutic intervention in Alzheimer's, which suddenly made people sit up and take much more notice of it. Second reason was a feeling that industry was not doing what it could or should be doing, and I'll come back to that point. And the third, again, which I'll come back to, is a real excitement about what I've seen in terms of the science going on inside industry and the opportunity to do something. So this slide is a slide that I showed when I joined GSK to try and introduce myself to some of the teams around the world. And what I did was put together some of the things I've been working on at UCL. So actually my initial background in research had been very much in human vascular physiology, infusing drugs intra-arterially, measuring blood flow and so on, and trying to understand the control of vascular tone. So that was a sort of bedrock technology, if you like. I got very much into things like cellular biology and cell movement, particularly in endothelial cells. I'd got involved with structural biologists, Neil MacDonald and others at Birkbeck, and we'd solved the crystal structure of a protein that we were interested in and made some small molecules against it. We had animal models of atheroma. We made a cardiovascular risk prediction tool which we were using in clinic, and we used very large databases, including the GPRD database, to try and address a question which was, does infection or inflammation cause a transient increase in cardiovascular risk? So there were some themes running through this about cardiovascular risk. The reason I put this down is when I put all these things down and I realized I was completely non-expert in almost every single one of them, again, it told me that interdisciplinarity, linking with colleagues, and actually taking advantage of the multidisciplinary organizations that I suspect you all work in, I believe is absolutely critical. And I've seen throughout my career individuals who sit in hugely powerful, hugely exciting, multidisciplinary organizations who never ever take advantage of that and stick with one thing. Now that may be fine if that's actually what your research demands, but if your research demands interdisciplinarity, you need to go out and seek it. And my experience has been you get a hugely positive response from people and learn a lot. So interdisciplinary research, I think, is important. So this leads me on to my last bit here, which is change. And when I look at my career, I made several changes of direction, and I think there was nothing more profound for me than the change I made to leave what I absolutely assumed was my lifelong career, which was in clinical academia, and to join industry. And if you'd asked me the day before I made the decision, literally the day before I made the decision, I would have told you that my career was going to be in clinical academia forever. And so what made me change my mind? I had actually very little to do with the pharmaceutical industry. I'd had no grants from the pharmaceutical industry. I'd had that 1 year of experience working as a postdoc funded by the Clinician Scientist grant. And I'd written quite a lot of negative things about how I saw the industry not really delivering what it needed to deliver for patients and being too focused on profit and not enough on applying the science to make great medicines. I got invited to join the Research Advisory Board of GSK, which was the group that overlooks all of the research activities of the company. Inevitably, you can't really dig into any detail because it's so huge, but I got to see the whole process. And it was a massive eye-opener for me when I was on the Research Advisory Board that the science was outstanding, it was truly interdisciplinary, and actually there was some great stuff and exciting stuff going on. So I was sort of primed, but I still thought my career was in academia. I had dinner one night with the then head of R&D, Tashi Yamada, who said to me, why don't you join us? And I said, it's not for me, my career is in clinical academia. And he said, what about if you come and head drug discovery? That's actually quite interesting. That's a difficult problem. What am I going to do? Am I going to spend the rest of my career criticizing what I saw as a failure there, or go in and try and do something about it? And almost overnight, I made the decision that I would join. I joined. I haven't regretted it for a day. And a number of things that I've found since making that decision. The first is you absolutely can't shy away from testing your idea in industry. And what I mean by that is, and I've sat on enough grants committees to know this is the case, there are people who spend most of their career dancing around the edge of the problem and getting recurrent grants to do so and never tackling it head on and saying, is it true or is it false? And you cannot do that in industry because you're going to make the medicine, it's going to go into the clinic, and you'll be right or you'll be wrong. And that, I think, is an attractive thing, for me anyway, in terms of testing ideas. I found outstanding science, and I've said this before, when I joined GSK, one thing that absolutely surprised me at the time was it felt a bit more like the public service than the public service when I first joined, because it was so big and so secure. But of course all of that's changed, and it's a very different animal now in terms of the way we think about the structure and of course the economic pressures on industry, and it's much more nimble, much more actually in many ways like a very focused academic organisation in parts and a very focused business organisation in terms of delivery, with much less of the sort of rather solid structures that I think were in place before. But there was no question that there was really outstanding science, and I think that has continued. I can say a word about why I think it's different now. I think there's all sorts of things that have happened, including a much greater focus on being externally orientated. About half of what we do is linked to the external world now, not totally internally focused. Publications, we had something like 20 publications in either Nature, Nature Medicine or Science last year. Get the publications out there, it gets you credible in terms of your academic peers. It also means that you can actually talk to people as equals from that science base. I also find huge entrepreneurial spirit in terms of trying to do something practical with a fixed sum of money. So I think industry is in a very different position. I'll come back to that now. And people often say to me, what do I miss from clinical academia? And I do miss things sometimes. I do miss the sort of banter that goes on across a multidisciplinary organisation and the interactions you can have with people way outside your field, although I do a lot of that in industry. Probably the thing I miss most is students. And actually it's the challenge, the inspiration, the excitement of super bright people asking bloody difficult questions, which I think are the thing that I perhaps miss, and that's one of the reasons why this year, in fact, we're going to have a big graduate recruitment drive at GSK. I'm sure others will do the same, to try and really refresh the organisation after a period of contraction and downsizing. So, let me lead to my final part, which is about keeping an open mind. I'm very well aware, as I said, that training is more structured now than when I trained. I'm also aware that there are certain routes that become impossible perhaps, or seem impossible, and some that are predefined. Others will be much more expert than I, and I can't possibly comment on the details of those things any longer, but I do believe that actually following what you believe is an exciting path is more important than what somebody else tells you is exactly the right path. I think you do need to take risks in this. And of course, when you take risks, there are no guarantees what the outcome is the other side. So there's risk associated with all of this, and I would encourage you to think about that. I also think, and John has already alluded to this, all of the grant-giving agencies, all of them, I believe, are totally supportive of young and new investigators. I think there's a big will to try and make sure that your independent careers get off to a start. I think that's a great environment in which to do it. I was horrified recently to hear in the US that the definitive NIH grants, the ones that really mark you out as an independent investigator, the average age of receiving one of those, I think, is something like 51 now, for the first time, which is just shocking. And we don't want to get into that situation. So let me now finish by talking a little bit about industry and careers, and try and maybe answer some of the questions that I know people have. So the first one is it a one-way step? Do you step out of clinical academia into industry and then never come back? My answer to that is a clear no. I think we've got examples of people transitioning. We've got people who've been in industry for a long time who are now in academia, and we've got people who do move back and forth. It's not as common as it should be. I've given you some reasons why I think you learn a different approach to research in academia, I think in industry. I think the interdisciplinary nature, the very project-focused nature actually is a very interesting thing for academics to get their hands on. I think the integration of chemistry and biology is much closer in industry than it is in academia. I'll give you a couple of quotes. One is from an academic, a senior academic that I recruited about a year ago. And I was asking him about this one-way step, and he said, it's very odd, he said, but the best job offers from academia came after I joined GSK. And I think that's probably true. The second one is from Caroline Savage, who some of you will know, who was Professor of Medicine in Birmingham and a very senior academic in the UK. And she said, this isn't something I would have, she's been with us probably for 2 years, I think now. She came initially on a sabbatical and then has stayed. So it's not something I would have predicted, but I'm closer to fantastic science than I was. I'm very focused on patient need and much more able to think about multiple diseases than I was when I was in my last post. Had I realised, I would have made the move sooner. So I think there are examples of people. Now, they're senior people, and I think the challenge for us and the challenge for the UK and the challenge for academia is to make movement much more flexible at a more junior level as well. Because I think there are things that can really benefit research training from spending a time in industry, and we need to make those moves very flexible. One of the problems is when people come in, there's a view that they won't be able to publish. And I've given you some examples. We do publish. We publish all of our clinical work, and we publish a lot of our basic science work. Everything actually, except for the molecule structure too early, because that's where intellectual property resides. So we publish a lot, and I recognise that publications are the currency of academia, and therefore we want to make that easy for people to do that. But I do think on the other side, academia probably needs to recognise some of the skills you get from within industry, which are not to do with publication, they're to do with integrated research thinking. Another question that comes up, is it bad money? And I think this is not a trivial question. The industry has had its reputational problems. It clearly has had a lot of issues over the past. And all I can say is, repeat really what Caroline said, I don't think I've ever been more focused on patient need, on applying science to try and do something which is important for patients, and thinking about long-term healthcare systems than I've ever been in my career. And I think certainly at GSK we've absolutely got values and principles which are driven by the need to be open, the need to be part of the scientific community, and the need to understand that the only reason the pharmaceutical industry can operate is because society lets it to operate, and therefore you have to be part of a really responsible societal interaction in order to allow this to happen. Is it second-class science? I think I've told you, and I can't say more than the papers that come out, it's absolutely not second-class science, it's world-class science. One of the things that still slightly depresses me sometimes is when I know that we've solved a problem that others are working on and we haven't yet published because we haven't secured the IP in an early stage of work, and I think we need to get better at doing that. It's world-class science. Is it all chemistry? Absolutely not. And what's interesting is not only is the biology important, the integration of chemistry and biology, which I've talked about, but the number of other things that we get involved with, ranging from what is the impact, for example, of the ENCODE publication or the understanding of epigenetics on the way we think about drug discovery and development. We have a lot of work going on there. What is the impact of large electronic patient databases on how we think about disease epidemiology, about disease progression, and about how our medicines can be used? And how do you pick up signals? How do you pick up a safety signal of a medicine from a large electronic database? Very, very interesting, difficult question, one that actually means you have to work with other industries. We've got some links with a company that actually detects signal detection for bank fraud and for intelligence surveillance. And many of you will know the moment you make one error in your PIN code when you put it into your bank card in some foreign country, the bank's onto you in 2 minutes. They know that something funny has happened, and, or some odd pattern of spending. Those are similar to trying to pick up signal detection for safety. So we work across disciplines. We have engineers, we have informatics people, we have geneticists, we have others. It's not just chemistry. And I'll come back to this question that ultimately in industry you can't shy away from the question, does your hypothesis work when you put it in the clinic? And that for me is what's attractive. In the UK, we have a unique position. We have still relative over-presence of the pharmaceutical industry. We have absolutely outstanding academia. We have a somewhat underweight biotech sector, but we're one of the few places that has a truly integrated potential for these sectors to work together. And I think there's a real chance there to do more of this. And it requires, I think, something around career development and something particularly about clinician scientists getting in, because we only really have 4 big questions in the industry. What am I going to work on? What's the target? What's the molecule I'm going to choose? Which is often a chemistry question coupled with clinical. What's the clinical experiment I can do that demonstrates that this has some potential? And what's the evidence I need to generate to show to society that this medicine works and adds value? All of those require input from clinician scientists at every stage. Thanks very much for your attention.